Hello everyone, I'm Mulle Mala, a forest volunteer. Today we're gonna learn about crime scene investigation of biological evidence. Here are the topics that we are going to learn today. Introduction. Crime scene investigation of biological evidence involves the collection, preservation, and analysis of biological material that can provide critical information in criminal cases. This type of evidence include blood, saliva, hair, tissue, and other biological samples. Now, what is biological evidence? Biological evidence refers to any biological material collected from the crime scene that can be analyzed for forensic purposes. This includes substances like blood, saliva, semen, hair, skin cells, and other body fluids. These materials are critical in forensic sciences as they contain unique genetic information, primarily DNA, which helps to identify the individual involving in a crime. Importance of biological evidence in forensic science. First one, linking individual, suspect and victim. Biological evidence plays a crucial role in establishing connection between suspect and victim. By analyzing biological tools such as blood, saliva or hair found at the crime scene, forensic experts can determine whether these samples match the DNA of the suspect. Individual identification. One of the most significant application of biological evidence is in individual identification. DNA profiling allow forensic scientists to create unique genetic profile from biological samples. This technique is instrumental in identifying unknown victim or confirming the identity of the suspect. Reconstructing crime scenes. Biological evidence is vital for reconstructing the event of crime. Forensic analysis of bloodstain pattern or the distribution of biological materials can offer insight into dynamic of a violent encounter, including the positions and actions of the individual involved. This reconstruction helps investigators to understand how the crime unfolded and can guide further inquiry and analysis. Fourth one, supporting criminal investigations. Biological evidence support the broader criminal investigation by providing tangible proof that corroborates witness statement or other types of evidence. The process of biological material can link a suspect to multiple crime scenes or establish pattern that indicate a specific modus operandi. They strengthen the overall cases and assist law enforcement in building more comprehensive understanding of the crime. Fifth one, exoneration of the innocent. Biological evidence is instrumental in exonerating individuals wrongfully accused or convicted of crime. Advances in DNA testing have allowed the re-examination of biological samples from past cases, leading to the cleaning of innocent individuals. The function of biological evidence highlight the importance not only in securing conviction but also ensuring that the justice is served fairly. The last point is time frame of event. Analyzing biological evidence can help establish a time frame event leading up to the following crime. For example, the condition of biological materials can indicate when a crime occurred, while specific biological markers may reveal the time and time of interaction between the suspect and victim. Understanding the chronology of event is critically for constructing a timeline that assists in crime investigation and provides content for the crime. Now, here are the types of biological evidence. They are blood, saliva, semen, hair, skin cells, urine, feces. Collection of biological evidence. Step 1, securing the crime scene and ensuring the safety. Secure the crime scene to prevent unauthorized access. Ensuring the safety of all persons, investigator, responder, etc. Access potential hazardous, biohazardous, sharp objects, environmental dangers. Wear protective gears, hand gloves, mask, cloths to prevent contamination. Maintain a detailed chain of custody for all evidence. Minimize the personal movement and avoid unnecessary contact with the evidence. Step 2, medical assistance and documentation. Providing medical assistance to the victim. If there is any injured person at the crime scene, the priority is move immediately to medical aid. Call emergency medical service to the scene of crime for victim stabilization and transport. Law enforcement or first responder officer should ensure that medical person do not disturb potential evidence unnecessarily. Second one, documentation of the crime scene. And third one, documenting injury and medical intervention. Fourth, collecting biological evidence. And at last, we need to maintain the integrity of the crime scene. Third step, initial scene assessment and evidence identification. Involve a systematic walkthrough of the crime scene to gain an overall understanding while avoiding evidence disturbance. Investigator visually identify potential evidence, particularly focusing on fragile or translucent materials like blood or trace evidence. Prioritization is given to collection items that are degraded or easily contaminated. Through documentation include photograph, videograph and sketching is essential to capture scene accurately. Environmental factor considered to prevent evidence degradation and safety precautions such as using PPE are maintained to protect both the personal and evidence while ensuring the chain of custody helped. Fourth step, documentation of the crime scene and evidences. Emphasizes the comprehensive recording of all aspects of the crime scene to preserve its original state of investigation and court proceedings. This state includes in taking extensive photograph, videograph, written notes of the scene from multiple angles and perspective. Every piece of evidence is documented before the collection with a detailed description of location, condition, and the relationship between one other evidences. Diagram and sketching are also created to provide an accurate representative of the scene. This is through documentation ensure that all the observation and evidence can be revisited and analyzed throughout the investigation while maintaining the integrity for legal proceeding. Now, the fifth step is collection of biological evidences. We already know the biological evidence they are blood, saliva, semen, hair, skin, urine, feces. Collection of biological evidence. First one, collection of blood. We are not supposed to consider every red pigment fluid as blood. We need to test each of them. Blood collection for forensic analysis involved drawing blood stains using sterile techniques to prevent contamination. A non-alcoholic solution is used to test the clean sites, ensuring accuracy. Alcohol test. Trained professionals handle the process, especially in postmortem cases where blood is the often drawn from major vessels. Second one, collection of saliva. Saliva collection in forensic analysis is a non-invasive process used to gather DNA or other biological evidence. It is typically done using the sterile cotton swab, which are generally rubbed inside the cheek to observe saliva. This method ensures a sufficient amount of epithelial cell which contain DNA. Collection of semen. The collection of semen in forensic investigation is vital for cases involving sexual assault. Semen is often collected from a victim's body or clothing as well as from the crime scene or object. Swabs are used to gather the samples from the area like vagina, anus, or mouth depending upon the case specificity. In some instances, semen stains are collected from the fabric or surfaces. Collection of hair. The collection of hair in forensic investigation is essential to cases involving in DNA analysis microscopic comparison. Hair can be collected from the various locations including the crime scene, victim, suspect or personal belonging. Forensic experts use tweezers, gloves to handle the hair stains, ensuring they are not contaminated. Both the head hair and body hair may be collected depending upon the cases. Collection of skin cells. Collection of skin cells for forensic analysis is done using sterile swabs or adhesive tape to gather cells from surfaces that the person has touched. These samples are placed in sterile container, labeled and stored properly to prevent contamination. analyzing the collected skin cells in the lab, allowing the DNA profiling, while can a link to individual to crime scene through touch DNA analysis. Maintaining the chain of custody is essential to preserving the integrity of the evidences. Collection of urine. Urine collection for forensic analysis typically involve a clean catch method where an individual provides a sample in sterile container to avoid contamination. In the cases observed, collection may be necessary. Once collected, the sample is secured, sealed, labeled, and stored in the appropriate temperature to preserve the integrity. Maintaining the chain of custody is essential to ensure the evidence is admissible in court. Lab analysis of urine can provide crucial information about the substance use of exposure. Collection of pieces. The collection of pieces in forensic investigation is vital for solving cases like sexual assault and environmental crimes. Packaging and preservation. Proper packaging and preservation of biological evidence are essential in forensic investigation to maintain the integrity of samples such as blood, saliva, feces. These procedures include using sterile or leak proof containers, sealing from securely, and labeling the critical information. Effective methods such as temperature control and contamination prevention are crucial. Additionally, maintaining a clear chain of custody ensures legal accountability. Adhering to these practices is vital for ensuring accuracy, forensic analysis, and supporting judicial processes. Laboratory analysis of biological evidence. First one, DNA profiling. Laboratory analysis of biological evidence, especially DNA profiling, is crucial in forensic science. This involves extraction of DNA samples from blood or saliva, following by amplification using the techniques such as PCR polymerase chain reaction. DNA profiling identifies individual by examining specific genome regions, effectively linking suspect to crime. It accurately enhances criminal investigation to provide a vital insights for legal proceedings. Second one, PCR, polymerase chain reaction. Polymerase chain reaction is crucial in forensic techniques used to amplify DNA from biological samples like blood or saliva. This process involves an extraction of DNA and cycling through detonation, annealing, and extension to replicate the specific region. PCR is vital for effective DNA profiling, specifically when sample quantities are limited. The third one is blood typing. Blood typing is a key forensic technique used to identify an individual's blood group based on the antigen present on the red blood cell. By mixing up both blood groups with the antisera, expert can determine the blood type like A, B, AB, O, and RH. This process helps in link suspect to evidence or exclude individual in crime scene investigations. While less specific DNA profiling, blood typing provides valuable information and serves as a rapid, cost effective preliminary scrolling and tool in forensic analysis. The fourth one is short tandem repeaters STR. Analysis is a crucial forensic technique for DNA profiling that examines repeated DNA sequencing. It involves extraction of DNA amplifying specific region using PCR and separating fragments to create unique profiles. This method is essential for linking suspect to crime scene in crime scene investigation and a paternity test. Fifth one is saliva testing. A saliva testing is a valuable forensic tool to analyze biological samples for various applications, including DNA profiling, drug testing, and detection of pathogens. Saliva contains DNA, enzymes, and other biological markers, making it suitable for forensic investigations. Sixth one is semen analysis. Semen analysis is a crucial forensic technique used in sexual assault investigation. It involves collecting semen samples, examining them for sperm motility and morphology. Extraction of DNA profiling using methods like PCR or STR analysis. This process helps to link suspect to cases, exclude individual, and provide essential evidence in a legal proceeding. Seventh one is hair analysis. Laboratory hair examination involve analyzing hair sample from a crime scene to identify or exclude individual. The process includes microscopic assessment of hair characteristic and DNA extraction for profiling when root are available. The techniques provides valuable evidences in forensic investigation laid in linking suspect to crime scene. Challenges in handling biological evidences. Handling biological evidences poses change like degradation, environmental impact, and time sensitivity. Samples can deteriorate from temperature, humidity, and contamination, compromising their integrity. Environmental factors such as sunlight and pollutant can worsen their degradation. Time collection and analysis are crucial to preserve critical marker and making careful handling essential for reliable results. Contamination. Contamination poses a significant risk in handling biological evidences as it compresses and the integrity and the reliability of the samples. Careful protocol and a sterile technique are essential to prevent contamination during the collection, storage, and analysis. Cross transfer between suspect and the victim. Cross transfer of biological evidence between suspect and the victim complicates forensic investigation by blurring the link between evidence and individual. This can lead to misinterpretation of results, making accurate analysis and contextual understanding essential in assessing the relevance of the evidences. In conclusion, investigating biological evidence at a crime scene is essential for linking suspect to crime scene and enduring justice. Addressing the challenges like contamination and degradation is crucial for maintaining the integrity of the evidences and enhance the reliability of the forensic investigation. In conclusion, investigating biological evidence at a crime scene is essential for linking suspect to crime scene and enduring justice. Addressing the challenges like contamination and degradation is crucial for maintaining the integrity of the evidences and enhance the reliability of the forensic investigation. Thank you for watching. If you enjoy the content, please like, subscribe, and stay tuned for more exciting videos. Your support means a lot.